This lecture will be a review of protein purification. Initially, we started with the DHFR protein, which we would later on purify from E. coli cells. So prior to beginning the experiment, these E. coli cells had actually been transformed so that they would contain the DHFR protein. And the way this was done was through addition of plasmid. And the plasmid contained DHFR, so when the plasmid inserted into the E. coli cells, the DHFR protein also became inserted into the E. coli cells. DHFR stands for dihydrofolate reductase, an enzyme that is critical for nucleic acid synthesis, for example, DNA synthesis. The protein DHFR has medical applications. For example, a decrease in DHFR can lead to inhibition of DNA synthesis. Essentially, a decrease in DHFR will lead to impeded growth and proliferation of cells, and this will ultimately lead to cell death. So in instances when you have an overgrowth of cells, you need to reduce the activity of DHFR so you can reduce the growing or proliferation of cells. There are certain medications that can be prescribed, and these medications can directly inhibit DHFR, therefore inhibit the ability of cancer cells and their ability to make nucleotides. This is done through competitive inhibition. One medication is methotrexate. Methotrexate can inhibit DHFR, therefore it is used to treat cancers of the blood, bone, lung, breast, head, and neck. In addition, methotrexate is commonly prescribed for treatment of rheumatoid arthritis and psoriasis. For the first half of the protein purification lab, there were many protocols that students completed. As mentioned before, the plasmid pDHFR had already been transformed or inserted into E. coli cells. pDHFR stands for the plasmid, which contained DHFR. An overnight culture was initially prepared. The overnight culture would need LB-AMP broth, 20% glucose, and of course, E. coli cells. LB-AMP broth contains Luria-Bertani medium, which is a liquid media, in addition to ampicillin, and mixed with the 20% glucose will provide some nourishment to those E. coli cells, which we are trying to grow, and then further on, we are trying to purify. Followed by an overnight culture, absorbance would be measured. Absorbance is also known as optical density, or OD. However, this terminology is not encouraged. It is more accurate to say absorbance than it is optical density. Absorbance was measured at 600 nanometers, which is the wavelength at which the spectrophotometer was set. Followed by the overnight culture and the absorbance measurements, a subculture was prepared. Taking into consideration what your absorbance would be for your original overnight culture, you may have needed to add an increased volume of LB-AMP broth, or you may have needed to add not very much LB-AMP broth. Whichever the situation was, your subculture that was prepared should have had an ideal full absorbance of 0.3. And in order to achieve a subculture with an absorbance of 0.3, you would calculate just how much LB-AMP broth you would need to dilute it with. Followed by the overnight culture, the absorbance measurements, and the subculture, you would need to prepare an uninduced SDS-PAGE sample. Followed by that, you would need to take some more absorbance measurements of your subculture, and then you would add IPTG in order to induce DHFR. Adding the chemical IPTG will induce the expression of DHFR so that the protein will be available so that you can subsequently purify it. After induction, students would need to prepare an induced SDS-PAGE sample. Followed by that, they would need to pellet, lyse, and freeze induced cells. So this encompasses all of the steps which had been completed so far. The remaining steps included separating the soluble from the insoluble, and this would be done using a syringe and cell fractions. Followed by that, there would be preparation of more SDS-PAGE samples, which include soluble fraction, insoluble fraction, soluble PAGE, and insoluble PAGE. The two PAGE samples, soluble PAGE and insoluble PAGE, include Emily buffer. Now we are getting to the purification of the DHFR protein. This purification was completed, or would have been completed, through affinity column chromatography. Chromatography is a technique that allows for the separation of individual components from complex mixtures. For our particular protocol, students would need to pour, wash, and equilibrate the column. They would then bind their sample to the column, wash the column some more, elute protein off of the column, and this technique would result in 4 samples: the flow-through fraction, wash fraction, eluate fraction, and desalted eluate fraction. Using these 4 fractions, 4 SDS-PAGE samples would be created. That is, flow-through PAGE, wash PAGE, eluate PAGE, and desalted eluate PAGE. Followed by that, students would quantify or determine the concentration of the desalted eluate protein sample. This would be done via nanodrop 2000 spectrophotometer. That measurement of protein concentration would be recorded for the lab report. The final step in protein purification is SDS-PAGE. Before I get to SDS-PAGE, I just want to review plasmids. So recall that a plasmid is a circular DNA molecule. Plasmids can occur naturally, and they exist outside of the chromosome. In the image below, you see the bacterial genome, and then you see plasmids that are outside of the bacterial genome, but still encompassed in that bacterial cell. So plasmids occur outside of the genome. It is only during heating and cooling that you can possibly insert them, and this is done through transformation. Plasmids are used as a vector in order to carry genetic information, as we did in the PGLO bacterial transformation lab, as was also done for us prior to our protein purification lab. Prior to our protein lab, PDHFR had been transformed. It had been inserted into E. coli cells for us to use in order to prepare overnight cultures. Recall that when bacteria take up this genetic information, this is called transformation. The plasmid, as I mentioned before, was PDHFR, which indicates the plasmid of dihydrofolate reductase. And further into the protocols, you will notice that DHFR was tagged with GST and histidine. And I just want to review spectrophotometry next. Spectrophotometry is a technique used to measure absorption, and this is done using a spectrophotometer. Specifically, we are measuring electromagnetic absorption. This measurement will indicate just how much light is absorbed through a given substance at a particular wavelength in nanometers. There are several units of absorbance, and they all mean the same thing. I typically like to use ABS, which stands for absorbance. Sometimes you may see AU, which stands for absorbance units, and oftentimes you will see OD, which stands for optical density. Although OD is commonly seen, it's not encouraged. It is not the most accurate terminology to use. And in our lab, recall that we did utilize a UV-Vis spectrophotometer, which encompasses the UV spectrum, or ultraviolet visible light. Some common types of spectrophotometers include a UV-Vis spec, which will measure absorbance, and a nanodrop 2000 spec, which will measure the concentration and purity of DNA, RNA, or protein. In our lab, we previously used the nanodrop in order to measure the concentration of DNA, and we also used it to measure the purity of DNA. For this particular experiment, we would have used the nanodrop to measure concentration of protein. Spectrophotometry versus spectroscopy. So recall that spectrophotometry encompasses many different instruments which can allow you to measure absorbance or the amount of light which passes through a substance. Spectroscopy, on the other hand, will allow you to measure the interaction between matter and electromagnetic radiation. There are several different types of spectrometers, which are different from spectrophotometers. One type is the IR spec, also known as FTIR spectroscopy. An IR will allow you to identify functional groups in liquid or solid chemical samples. Sometimes these chemical samples may be unknown. So an IR will allow you to determine if you have a carbon-hydrogen bond, if you have a carbon-oxygen bond, if you have an H bond, if you have a C-triple-O bond, etc. NMR spectroscopy will allow you to identify just how many carbons and hydrogens you have in a compound in addition to their arrangement. So for instance, if a particular carbon has 2 hydrogens attached or if it has 3 hydrogens attached. IR and NMR together will allow you to identify a chemical structure which may be unknown. So these are types of spectrometers. Also, they can be sometimes identified as spectroscopes. Sometimes they are identified as spectrophotometers, which is not very accurate, but you will see these terms used interchangeably a lot. For example, UV-Vis, that's a spectrophotometer, although in certain literature, it's just called a spectrometer or a spectroscope. And these are used interchangeably, but it's important to realize that they are 2 different techniques, essentially. And then there's a few other types of spectroscopes, Raman spectroscopy and mass spec. And so in the image on the bottom left of the screen, that is an IR or FTIR spectroscope. And then on the right-hand side of the screen, that's an NMR spectroscope. Next, I have a reference chart, and this is for an FTIR. It has a list of different types of bonds and elements, essentially. Depending on the peak intensities, which you will see towards the bottom, you can determine just what chemical bonds you have in your sample. And this would be primarily for the FTIR. Next, I'd like to go over chromatography. Chromatography allows for the separation of individual components from complex mixtures. There can be many different types of chromatography. For example, column chromatography. For the protein purification lab, we would have utilized a column very similar to the one on the bottom left of the screen for affinity column chromatography, and this would have allowed us to add reagents, centrifuge, in order to bind and then elute the protein off of the column. There's also gas chromatography mass spec, also known as GCMS. This does utilize machinery and a computer in order to perform GCMS. There's also HPLC, or high performance liquid chromatography, which also requires an instrument and a computer. Thin layer chromatography, on the other hand, can be done manually, very similar to column chromatography. However, for TLC, you do require a silica gel sheet in order to pipette chemical solutions and then see the separation of molecules, particularly under UV light. A different example of column chromatography would be the image on the right, which is size exclusion column chromatography. And these are a bit larger in size, these columns, and these are used to separate molecules that are of different sizes. For example, a common experiment would be separating vitamin C from hemoglobin using column chromatography. Just to recap recombinant proteins are artificial proteins, and these are created through a technology known as recombinant DNA. So recombinant DNA includes different types of DNA from various organisms, and the DNA is combined into living cells, and then from that DNA, the gene codes for a particular protein, which is an artificial recombinant protein. So DHFR that we utilize for the protein lab would be known as a recombinant protein. And to make recombinant proteins, first you would need to isolate the gene and then clone it into an expression vector, commonly called a plasmid. Most recombinant proteins typically originate from humans, however, they are expressed in other organisms prior to therapeutic use. These recombinant proteins might be isolated from humans, but they are inserted into bacteria, commonly, or sometimes yeast or animal cells. Examples of recombinant proteins that we use for therapeutic use may include insulin, tumor necrosis factor, and relaxin. The final portion of the protein purification lab would encompass SDS-PAGE, which is a type of electrophoresis, very similar to agarose gel electrophoresis. SDS-PAGE is sodium dodecyl sulfate polyacrylamide gel electrophoresis. This type of electrophoresis will allow you to separate bands of protein based on size. It is similar to agarose gel electrophoresis in that you can separate bands of macromolecules based on size. This particular electrophoresis is specifically for protein, whereas agarose gel electrophoresis is for the separation of DNA based on size. On the right-hand side, you can see a cassette, which includes a SDS-PAGE gel. It's a very thin gel, and it comes pre-packaged from Biorad. This particular gel has about 12 wells, and the protein samples, which may have included, wash page, flow-through page, induced page, uninduced page, all of these samples would have been pipetted vertically into the very thin gel, which itself is standing vertically, in addition to a marker, and you can actually fit 2 prepackaged gels on either side of this green structure. And there are 2 of these structures which can fit into an electrophoresis chamber. In the next image, you'll see the electrophoresis box in the middle. As I mentioned, you can have 2 cassettes in a given structure, and 2 of those structures will fit into the electrophoresis box. Followed by that, you will attach the lid, and then you will connect the electrodes to the power source. One thing about SDS-PAGE is that it only takes sometimes 15 minutes to run the gel, whereas agarose gel electrophoresis can take about an hour to separate bands of DNA. Whereas for SDS-PAGE, you can ideally just utilize it for 15 minutes in order to separate bands of protein. And on the left and the right, you'll see the SDS-PAGE gels, which have a purple stain. And then you'll see on the very right-hand corner in the top, you'll see the marker on the leftmost well of that gel. And then on the very bottom right, you can see the dark purple loading dye, which was added to all of the samples prior to pipetting. And once all the samples are pipetted and the buffer has been added into the chamber, that would be when you would begin the run. Just an overview of SDS-PAGE. Initially, you would have your protein samples and the marker loaded into the vertical system. And you would also have buffer filled up way past the protein samples. Once you begin the SDS-PAGE run, the samples will migrate downward, and upon completion, you will stain the gel, or would have ideally stained the gel. And that would be later for analysis. On the right-hand side, you can see the chamber, the electrodes, the structure which can house 2 of the prepackaged gels. And then finally, 3D protein structures. So you have the primary structure on the bottom. You have secondary structure, which can include alpha helices and beta pleated sheets. You can have your tertiary structure in the middle, and then quaternary structure. And you will be familiar with all of these structures upon completion of the bioinformatics exercises. And that's it for the protein purification keynote.